Oh, I just want to say hi, everyone. Welcome to our ICICA webinar. Feel free to leave your message, questions in the Q&A box, and then we will start shortly, and we'll just wait for 1 minute for everyone to join in. Yeah.
Maybe we can start.
Yeah, I think we can start now. Yeah.
Okay. Hello, everyone. My name is Dali Wang. I'm the host for today's seminar. Before we start the formal presentation with the speaker, let me introduce the ICSA. ICSA is the International Chinese Statistical Association. Currently has over 2,000 active members. ICSA published 2 scientific journals, Statistical Seneca, Statistics in Biosciences. ICSA co-sponsored several annual conferences. For example, ICSA Applied Statistics Symposia, ICSA China Conference, ICSA International Conference, etc. So we have this webinar series is focused on career development in pharmaceutical industry. So we have 2 sections. One is technical trainings. Today's presentation, Causal Inference in Pharmaceutical Statistics, is part of our technical training. We also have another section is soft skills, leadership, and development. We have 9 committee members to support these activities for webinar series. We started this webinar series last year. Today's series will last about 45 to 50 minutes. And if you have any questions, you can put in your question in the Q&A. The speaker will answer your question at the end of the talk. Before the presentation, let me introduce the speaker, today's speaker. Today's speaker is Dr. Yixing Fang. He is the Director of Medical Affairs and Health Technology Assessment Statistics in AbbVie. He is also a research fellow of AbbVie Community of Science. His research interests include causal inference and machine learning with applications in clinical trials and real-world studies. He co-edited a book called Real-World Evidence in Medical Product Development in 2023, and authored a book called Causal Inference in Pharmaceutical Statistics in 2024. He is a co-member in 2 ASA Biopharma section, statistical working groups, real-world working group, and neuroscience working group. And he served as the associate editor for Statistics in Pharmaceutical Research. Without further ado, I'll turn over to Dr. Fang. So Yixing is yours.
Thank you, Deli, so much for inviting me, and thank you, Deli, for a nice introduction. So So today's talk is about causal inference in pharmaceutical statistics. Actually, it's a course 101. So Deli asked me to make it simple for younger generation who want to know something about causal inference in pharma industry. Okay, so this book, the materials for this webinar are from the book Deli just mentioned, with the same title. Okay. And this is outline. So we start with RCT, and then we go to non-interventional studies. And we mention briefly about semi-parametric statistics. This is the theory behind many innovative methods. Working in the pharma, if I were asked for advice about learning pharmaceutical statistics, my advice would be read the guidance. It is the masterpiece for us all. So this is similar to like as a mathematician, we always want to learn, read Euler. And then in terms of guidance documents, there are two series, very popular one. There are others type of document, but usually in pharma, we are interested in reading these two series of guidance document. One is ICH document, another is FDA guidance document. For example, for ICH E9R1, so this is very popular guidance document you see from the title. So how do we define estimand? How do we conduct sensitivity analysis in clinical trials? And for FDA guidance, you can search online, so fda.org. And for example, one recent FDA guidance is called Real-World Evidence Considerations Regarding Non-Interventional Studies. So if you are doing RCT, so you can start with ICH E9R1. If you are doing observational study, you may start from this guidance. And then, of course, all the guidance documents are related to each other. There are many others. So here are just two examples. So for ICH guidance, you can go from here, ich.org. So you can see many, you can find many guidance documents. And this is one example of ICH in R1. So you can see how important it is. So this document point out the central questions for drug development and licensing are to establish the existence and to estimate the magnitude of treatment effects. So estimating treatment effect is our main goal. So how the outcomes of treatment compares to what would have happened to the same subject under alternative treatment. So you will see that this ICH R1 document, they didn't use the term called potential outcome, counterfactual outcomes, because in literature, somebody call it potential outcomes, somebody call it counterfactual outcomes. So they don't want to use a name, potential outcome or counterfactual. They just spell out, they just give a definition of a potential outcome. So for each patient in potential world, so you have two potential outcomes. Treatment 1, treatment 0, you have two potential outcomes. So this only exists in our imagination. So we need to imagine same patient can take two different treatments. And this is the causal thinking we are going to focus on today. And now you look at the FDA guidance, you can from fda.gov. There are so many, you can search for keywords and then you can find. So this is one example. This guidance only have 8 pages. But if you search causal this keyword, you will see 10 outputs. That means in the same document, 8 pages, you can see 10 matrices of causal. That's why causal thinking plays a key role in both documents, in ICH documents or the FDA documents. So in ICH documents, you don't see causal word. They don't call it causal. They just spell out everything. And in FDA guidance, they dare to call it causal. Okay. There are two common types of studies in pharma. One is RCT, randomized controlled trials. The other is non-interventional studies. You can say observational study, sometimes real-world studies. So there are different names. So today we focus on RCT first, and then we move on to NIS. You'll see that RCT might be maybe considered as one special case of NIS. You'll see. Okay. There are so many study designs. So RCT and real-world studies include pragmatic RCT, open label, for example. And then observational study, single arm with external controls. And there are so many challenges, missing data in the current events, time to event outcomes, time-dependent confoundings. So there are so many things we need to handle. That's why we want to use the causal inference thinking. But we only have 40 minutes today, so we cannot go very deep, all different kind of study designs. We are focused on one ideal RCT and one ideal cohort study. And then you see the idea of a causal inference thinking, and then you can generalize the ideas to other study designs. Okay, let's look at this wiki. So wiki is fun to read when you want to do some research, although you cannot cite it as a reference, but always a fun thing to start. So if you search this Rubin causal model, you will see this very nice first paragraph about potential outcome. The Rubin causal model is an approach to the statistical analysis of a cause and effect based on the framework of potential outcomes, named after Rubin. The name Rubin causal model was first coined by Holland, very famous statistician. The potential outcome framework was first proposed by Neyman in his 1923 master thesis in his 20s. So genius. Okay, so Neyman, we are familiar with him. He proposed idea about hypothesis testing, null hypothesis, alternative hypothesis, type 1 error, type 2 error, confidence interval estimate. Actually, he also proposed the idea of a potential outcome. But Neyman proposed the potential outcome in the framework of RCT, randomized studies. And then in 1970s, Rubin extended it to a general framework for both observational study and the RCT. So in pharma, people think, okay, we are doing RCT, we don't need a potential outcome, we don't need causal inference thinking. But actually, that's wrong. So people think, okay, only in the real world study, only in the observational study, only in the non-randomized study, we need causal inference method. But that's misunderstanding. Potential outcome was proposed first for randomized studies. Now let's look at RCT. RCT, we have to apply two common techniques. One is randomization, one is blinding. So randomization want to cut this confounding issue. And blinding needs to cut this indirect causal effect thing. So if you apply randomization, you apply the blinding, the study is a gold standard. Okay. Now there are two worlds. One is potential world. Each patient or each participant or each subject is wearing two hats. So if you are comparing two treatments, if I are comparing 3 treatments, those 1, those 2 with placebo, and then each patient is wearing 3 hats. But now for simplicity, we only compare treatment 1 with treatment 0. So each patient, in your imagination, in potential world, each patient is wearing 2 hats. So if we treat it by 1, what if? If we treat it by 0, what if? There are 2 what if outcomes, so-called potential outcome. But in real world, only 1 copy of the 2 potential outcomes is observed. So each patient is wearing only 1 hat. Now we have to connect these 2 worlds, right? Otherwise, nothing you can do. So data is useless. So we want to use the real world data you observe in the real world to infer, to make a causal inference about something in your imagination in real world, in potential world. We need to make assumption. So in RCT, assumption is easy. So if a patient is treated by one, then the observed outcome is equal to the copy, one copy of two potential outcomes. So corresponding to treatment equal to 1. So one of the two heads. But if in real life the patient is treated by treatment 0, the observed outcome is the same as one of the other copy of the two potential outcomes. Okay, so this is assumption. So there's no human error. So they are not like a mislabel something. They are not something else. So we have to make some assumption in order for us to make causal inference from real world to the potential world. Now let's go to the definition of estimand. So usually people forget about estimand. So they jump to methodology. They forget about what they are going to do. So estimand is something we are going to do. So if you are writing a SAP, a protocol, something, first thing first, you need to define what we are going to estimate. Estimand is actually in pharma is treatment effect. Okay, so you see the definition of estimand in this document, guidance document. A precise description of the treatment effect, reflecting the clinical questions posed by the trial objective. Of course, for the particular trial, you are interested in certain treatment effect. And that treatment effect is the estimand. So people are scared about estimand. So okay, I often hear people in pharma say, I hate estimand. But actually estimand is so important. If you don't know what you are going to estimate, you don't know how to estimate, right? So, and then this estimand summarize at a population level what the outcomes will be in the same patient under different treatment conditions being compared. See, in this guidance document, the estimand is defined using the terminology of potential outcomes. That means no matter what kind of randomized, non-randomized, or whatever study design you are doing, you cannot avoid the causal inference thinking using the potential outcome because the estimand is defined in terms of potential outcome. But this document don't call it potential outcome because some other names, some counterfactual outcome and so on. So this document just spell out to avoid confusion, to avoid like two schools of causal inference, people fighting against each other. So they want to avoid that. So they just define this potential outcome using plain language. What the outcomes would be in the same patient, but under two different treatment conditions. So that's the idea of potential outcomes. Okay, now we want to take a deep dive to the definition of estimand. A lot of things to learn for this simple sentence. From this sentence, you'll see that the definition of estimand has 4 attributes. You need to have 4 of them together to define estimand. Otherwise, you cannot define estimand. So you need to have, see, population level summary. You need to have a summary like a mean difference, proportion difference, restricted mean survival time difference, odds ratio, for example. You need to have some population summary. And you need to have one outcome variable. You need to have a population of patients. You need to define 2 treatment conditions in order to have some comparison. Of course, it's okay to work on single arm, but single arm only one treatment, and then you just describe it. You don't compare. If you don't compare, there's no hypothesis testing to tell if they are different and so on. So usually we compare treatment with a placebo, or we compare treatment with standard of care, or we compare our treatment with the treatment proposed by the other companies, for example, also active. So there are always 2 things you want to compare to make decision which one is better. Okay. So each estimand definition, you need to have 4 attributes: population level summary, outcome variable, population, and the treatment variables. Otherwise, something is missing. Okay, you need to have 4. Now let's give you 3 examples. One is continuous outcomes. So let Z be the treatment assignment, 0, 1, 0 or 1. 0 you can think about as placebo and 1 as the investigational treatment. And then Y is continuous outcome variable. So there are two potential outcomes, Y1, Y0. And P is a population. Usually it's defined through a set of inclusion criteria and a set of exclusion criteria. And now we define we use the population level summary is the population mean difference. So you have 1, 2, 3, 4. You get 4 attributes, and now you can define estimate. Estimate is the mean of potential outcome with treatment 1 minus mean of a potential outcome with treatment 0. And this theta star is the estimate. Okay, because we define this estimate according to the definition. We don't like jump from nowhere and then we define, we just, or you forget to define estimate. So don't forget to define estimate according to the definition of ICH. Okay. Now let's look at binary. Binary is like a yes or no, there's or not, and success or failure, response, non-response. So binary outcome we usually using the proportion difference. So you look at the two proportions and then look at the difference. But for 0, 1 binary outcomes, proportion is equal to the expectation actually, right? Because 0, 1, expectation of being 1 is equal to proportion of being 1. They are the same. So proportion can be written as proportion. Proportion difference can be written as mean difference for the 0, 1 binary. In survival analysis, in oncology, people are interested in time to event outcome, like a survival outcome. So survival outcome usually is having right sensory issue. So you cannot forever, you cannot follow a patient until die, right? Death. So usually you have to stop at a certain point. So that's right sensory. But actually underlying the right sensory. So if there's no sensory, so you can observe the survival outcome. The survival outcome is a continuous outcome if there's no sensory. But because of sensory, usually we fix a time point, like 2-year survival rate. So you can look at the survival rate difference. If you choose using the survival rate as population level summary, you compare the survival rates. So actually it is a binary outcome, survival or not, death or not. So it actually can also be written as expectation. So the other very popular population summary is called restricted mean survival time. Because the sensory, you cannot observe forever, so you have to stop a certain point. So if, for example, you are interested in restricted by like 1 year, and then within 1 year, how long the patient can survive. So this is restricted mean survival time. So it's just a minimum, the underlying survival outcome and certain fixed time point. So, okay, you can see that restricted mean survival outcome can also be written as expectation, mean difference. So you can put the 3 types of outcomes, continuous, binary, time to event. We can using the same definition of estimate, right? That's the mean difference. Very beautiful. The foundation is very beautiful. Okay. For continuous outcome, for binary outcome, for time to event outcome, you can using this expectation the mean difference as population summary to define your estimate. So we call it average treatment effect, ATE. So you hear ATE is the estimate we are going to estimate. Okay, and this expectation is taken over the population. So we have 4 attributes. So all the 3 types of outcomes, we use the same definition of estimate, mean difference. Okay, now we have 2 estimates. One is the causal estimate, another is a statistical estimate. Why people have to to confuse us by introducing two types of causal estimate. The reason is in potential world, there are two copies of potential outcomes, but in real world, only one copy is observed. Although the estimate is defined beautifully, you cannot observe both of them. It's useless. So we need to we need to do this identification. Identification problem is the most fundamental problem in causal inference. If one estimate is able to be identified, that means the estimate is estimatable. So we are able to estimate it. If somehow you cannot identify it, that means you need to make actual assumptions to make it be estimatable. Otherwise, using unlimited data, you can still cannot estimate it, right? So we want to, using limited data, we want to estimate the estimate. We need to translate, do this translate. So causal estimate is estimate defined in terms of potential outcome. That's ICHE R1 definition. And in our mind, when you were a student or you are still a student right now, so you might think, okay, estimate is some parameter in terms of observed outcome. But that's underlying assumption. The underlying assumption is the one we just mentioned, right? So we need to connect two words. So that's the assumption. Yeah, that's the assumption we make. Without this assumption, that means something wrong. Nothing you can do. So we need to make this assumption. Okay. The assumption is a so-called consistency. So the real world had to be one copy, one version of the potential world. So it had to be consistent with one copy, one version of the. Otherwise, it's inconsistent. There's no bridge to connect the two worlds. That means something is wrong. So you need to make assumption to build a bridge between two worlds in order to estimate. So we need to do this identification. I call it translation. We translate causal estimate to statistical estimate. Now let's look at RCT. It's very easy. So we are going to introduce to you two methods, but in RCT, these two methods are equivalent. They are complete the same. But in non-randomized studies, these two methods are different. That's why we have two methods, fundamental methods to estimate. But now in RCT, they are the same. You will find it out why the same. Okay. So theta star is the causal estimate we just defined according to ICHE R1 definition. And now we give the condition of treatment assignment because of randomization. So due to randomization, you can do the conditional, unconditional, they are the same because potential outcome is independent of randomization. Okay. But now we made assumption called a consistent assumption. So if Z equal to 1, observed outcome Y is equal to 1 copy of a potential outcome. That's why we can get rid of hat. So B. And same thing for the second term. Given the in real world is assigned to treatment 0 and the observed outcome is equal to one of the two copies. So that's so-called consistent assumption. So I put the reason here. Equality A mean because of randomization, equation B holds because of consistent assumption. And now we're done. Because at the beginning, we need to rely on potential outcome. But now, after identification, Y is observed, Z is observed, and then we are able to estimate it. So because this is a conditional mean, we use a conditional sample mean. So imagine you have two randomized arms, arm 1 and 2. So we use the sample mean in the arm 1 to estimate this population mean, and then we use the sample mean in the arm 0 to estimate this population mean. See, this is just the t-test and so on. You are able to do that, okay? Now let's look at the other method. The other method is called so-called IPW, inverse probability treatment weighted. So now let's look at A and B. Causal inference, causal estimate are the same. It's the same. But now we look at this equation A. Z is randomized treatment assignment. and y is potential outcome. They are independent. So the expectation of a product is equal to product of expectation. But you look at the first term here, it just the definition of proportion is equal to expectation of indicator. That's why the equation A holds. Now let's look at equation B. I means indicator function. So z equal to 1 is non-zero, otherwise 0. So if z equal to 1 non-zero, consistency assumption guarantee observed outcome y is equal to one of the two copies. And same thing here. Okay, so now we are done. So if you get this are done because z is observed, y is observed. So this is so-called statistical estimate. And now expectation, you can use a sample mean. Okay, now it's the whole sample, two arms combined together from I to 1 to the n. But now look at the denominator. It's the proportion of patients being assigned to treatment 1. So if you are doing 1-to-1 randomization, it's 1/2. So, but now you can use the number of patients in treatment arm 1 divided by the total sample size. So this is the proportion. But you see that this n and n cancel out. So these two estimates are the same. So that's why in RCT, we have two methods to do identification. But it gives us two equivalent versions and gives us two equivalent estimators. That's why RCT doesn't matter. But in non-randomized study, you'll see that two methods, you will see soon, these two methods will be different. Now, before we move on to non-randomized, we want to challenge you. So this is the RCT causal diagram. So Z is randomization, and Y is outcome. And what's X here? X is the baseline covariance. But why we need to consider baseline covariance in RCT? Simple answer. According to this FDI guidance, you look at this title, FDI guidance, adjusting, just new 2 years ago, adjusting for covariance in RCT. FDI said, adjust for covariance will improve statistical efficiency for estimating and testing. It just increased the power, reduce the variability, reduce the standard error. So we want to improve the efficiency. That's why we want to adjust for covariance. Okay. We are going to come back because RCT is one special of NIS. So when we have a general idea about NIS, we come back to this. But here, I just pose this as a question to challenge you. Okay, we have a causal estimate, we have a statistical estimate, and then we have unadjusted estimator. Just compare the sample mean. Okay, we can show that without covariance adjustment, the unadjusted estimator is consistent. Consistent means if the sample size goes to infinity, this estimator will go to the true value of estimate. Here are two questions for you. With covariance adjustment, is the adjusted method still consistent? We hope the answer is yes. And the second question is, how do we find efficient? So how do we find the best method? Who is consistent? And who has the smallest variance? So this is the goal FDI challenge us as a statistician to find a certain method to achieve this optimality. We want to find an estimator which is consistent and smaller variance. So in small sample world, so some of you are PhD students, you are doing qualified exams, you will see some problems about UMVUE. Minimum variance, uniformly minimum variance, unbiased estimate. So you hope the estimate is unbiased. You hope the estimate is having smaller variance. But that's for the small sample thing. So only for like a very spatial distribution, you are able to do like to achieve the CR, the Cramer law lower bound. Okay. But now similar, but not exactly the same, but similar. We hope the estimator is consistent, and we hope the estimator has the smallest asymptotic variance. So this is a larger sample theory. UMVUE is a small sample theory. Okay. Now we go to NIS. Again, NIS could be complicated, but now we focus on ideal case because we only have 15 minutes left. Okay, let's see if how far we can go. Okay. Now let's look at a perfect cohort studies. 2 cohorts, but non-randomized. Okay, no randomization, no blinding, open label. Actually, okay. And then 2 potential outcomes. The same thing. According to estimand definition, we are using the potential outcome language, and then 3 type of outcomes: continuous, binary, time to event. All of them can be written as mean difference. Okay. But now you see this causal diagram is a little bit different. The difference is, or now we don't using the Z, we're using A here. A means action, intervention, like treatment. Okay. Before you use Z, Z is randomization. But now we have a covariance also impact treatment. So this covariance is called a confounder. So this covariance is related to both treatment and outcome. Okay. Now we want to adjust for covariance. We want to adjust for confounder for 2 purposes. One purpose is, as FDA guidance say, we want to adjust for covariance to improve efficiency. The other thing is confounding bias. Without adjustment, the confounding cannot imply causation. The association cannot imply causation. But after careful adjustment of confounders, to some extent, you are confident that association implies causation. Okay. So now again, we have 2 worlds. One is the potential world, everyone have 2 heads, and real world only have 1 observed outcomes. And then we need to do the same thing. We need to translate the causal estimate to statistical estimate. But now we need to make more assumptions. In RCT, only 1 assumption, consistent assumptions. But in NIS, we need to make 2 other assumptions. The other assumption is called, you can say, no unmeasured confounders. All the confounders are measured, included in X. And then you have the positivity assumption. That means 2 arms have overlap. If 2 arms are well separated, there are no data, no common data. Like common data means within each level of covariance, you have data from arm 1, you have data from arm 0. So if given certain level of X, all the data go to 1 arm. That means positivity violation. That means nothing you can do. You have no data to help you. That's why we need to make 2 assumptions. But this 2 assumption is like a tug of war. So they are competing against each other. So if you look at the no unmeasured confoundings, you want to have as many confounders as possible. But having more confounders will increase the possibility of positivity violation. Because the more covariance you consider, more likely you can find a level of X such that all the data go to 1 arm. Okay, so you have to think carefully. You cannot have too many variables. You cannot have too few variables. You need to have a compromise. So that thing, we need to think hard about sensitivity analysis. That's why the ICH ENR1, you look at the title. Title have 2 key terms. One is estimate, another is the sensitivity analysis. Okay, we need to do sensitivity analysis to see how likely these 2 assumptions will be satisfied. And then if they are violated, what will have happened? So this so-called sensitivity analysis. Okay, now we need to do these 2 translations. Okay, I put formula here. So you can read it carefully. I put the reason here, A, B, C reason here. You can understand that, try to think about how to prove it. So you had, in your life, you had to do it this one time, at least one time. I did this like 20 times, 30 times. So every time you do that, you will think the theory is so beautiful. You need to make assumptions. You cannot get rid of any assumptions. Those are the least set assumptions. You cannot make more assumptions. You cannot make a few assumptions. So you have to make 3 assumptions. And then look at this one. This one is very easy. If we look at this, it's just a regulation function. It's conditional mean of y given a and x. A is treatment, x is confounders. So everything are observed. Y, A, X. So we're done. So this is one type of identification. And this is the second method. You have reason A, B, C, D here. So you need to think about yourself. So why? Why this holds? Why this holds? You need to prove once, at least once in your life. And then you can get this another version of a statistical estimate. Now to summarize, 2 methods to identify the causal estimate. Give you 2 versions. Actually, they are equivalent. You can prove that they are equivalent. They are, in theory, they are the same, but they just look different. So one thing is according to this regression function. One thing you look at denominator. This is our old friend, propensity-score function. So this one is a regression function, also our old friend. So you see that one is a regression function, one is a propensity-score function. Okay, now we introduce some notation. We're using Q to define the regression function of conditional mean of y given treatment and covariance. And propensity-score function is actually the probability of A equal to 1 or 0 given covariance. And then the estimate can be written as Q or Z. So there are 2 equivalent versions of a statistical estimate. If you happen to like this version, you can propose an estimator. If you happen to like this version of estimate, you can propose another estimate, another estimator. So there are 2 fundamental methods. That's why we had 2 methods, and then at the end of the day, they are kind of together. They are kind of the same method. Okay, let's see this. If you are interested in doing regression function for y and x and a, you can run a linear model for continuous outcome. You can run a logistic model for the binary outcome. You can run a Poisson model for the count data. You can run the whatever Cox model, whatever model for the time to event outcome, for accelerate failure time model for the time to event. You can run any model. And now you just plug it in. So you have a regression function, you plug in, you're done. Okay, now for this propensity-score. So usually you run logistic regression model, but actually you can run a decision tree model. You can run a neural network model. You can run a support vector machine. You can run the random forest boosting. Any model you can run, and then you can get estimate. You plug in, done. So 2 versions of estimate give you 2 classes of estimators. Okay. But after you get the point estimate, you want to get the standard error, right? Otherwise, you are unable to do the statistical inference, get the confidence interval estimate, get the p-value. Now, how do we get standard error? An easy method is a bootstrap. So without thinking about the statistical formula, you just do the bootstrapping. You do the bootstrap, you get 1,000 results, and then you get the standard error or confidence interval estimate. You're done. You get p-value, and so you're done. But in theory, we want to see the formula. So actually, the theory is called M estimation. But in pharma, we are using SAS. So in pharmaceutical, like AV, so we are using SAS to do the causal inference. So this is SAS PROC called the causal treatment. Causal TRT means treatment, causal treatment estimate. So using this SAS will give you everything. You give you either you give you this method or give you that method, give you p-value, give you confidence interval estimate, give you everything. This is the SAS code. SAS code, you have two model statements. If you specify this model, you get the first method, regression method. If you using the second model statement, you are looking at propensity-score method. Two statements, you give two methods. So SAS make everything very easy. You just plug in data and then get the result, get everything. But you look at title here. 1 plus 1 equals 3. You have one statement for outcome, one statement for propensity. 1 plus 1 equals 3. Why? Because you are curious. If you specify the first outcome model, you get first method. You specify the second model, you get propensity-score method. How about let's try this game? We specify the both model. Both model you give you one very nice method, so-called augmented IPTW. That's why 1 plus 1 equals 3. So two model statements give you 3 powerful methods. So this so-called doubly robust methods. So if a propensity-score is valid, your estimator is good. If outcome regression model is valid, your estimation is good. But these two methods are single robust method because you bet everything, the successful, whether the study, you spend $1 million on a clinical trial, and then you depends on how good your model is. But those model only exist in statistician mind. The model does not exist. All models are wrong, but some are useful. So we want to make sure if one of them is correct, and then the estimate is correct. This is the so-called double robust method. So that's why if you specify the two models, you increase your choice, your probability 2 times higher to be successful, to be accurate, okay? So this is so-called the double robust method. Now let's go back to RCT. So you say, okay, I'm doing RCT. I don't like those fancy methods. But actually, RCT, you can still use those methods because covariance, according to FDA guidance, we can adjust covariance to improve accuracy. But sometimes nothing is perfect. You run an RCT, you hope 2 arms are balanced. But in reality, 2 arms are not perfect balance. At that time, you want to adjust covariance for 2 things. For increasing the efficacy, the other is improve, adjust for the imbalance. So you can do a lot of things. But now, as I mentioned earlier, you are not satisfied by the linear model because the linear model is too naive, right? Too simple. You want to think about machine learning methods, AI methods. That's why nowadays we need to combine both. Machine learning and causal inference together, it will be very powerful tools, you will see. Okay, but in also, but machine learning people are worried about overfitting, like a lot of things people worry. But actually, don't need to worry because the theory behind the machine learning and causal inference combination is very solid now. The theory is called semi-parametric theory. So if you are interested in semi-parametric theory, there are a lot of nice books to read. One is Bickel's method, Bickel's book, but this is too hard. I don't recommend it. When I was a PhD student, we were using this book as textbook. It's like headache. And now you have Ciatis' book. It's much easier to read. But that one is in missing data framework. But now you have a causal inference book, What If, by Robbins. So this one you can learn causal inference and semi-parametric at the same time. And then this target learning is including machine learning and causal inference and semi-parametric together. And now this is dynamic treatment regime is even harder. So you want to look at the dynamic treatment like time-dependent confounding and so on. So all these books are good, much better than my book. My book just want to talk about causal inference, machine learning in terms of a pharmaceutical point of view. So we are doing clinical trial, we are doing observational study, we are doing intercurrent events, missing data, and so on. So we are going to using that one, and then we are how to relate to this FDA ICH guidance with causal inference. That's what we are doing today. Okay. So and then a lot of application. You might think, okay, too easy because we only have 15 minutes. Excluding the introduction, we only have 45 minutes today. So I only give you the ideal RCT, ideal NIS, but actually there are a lot of challenges. Your external control and longitudinal, how to handle missing data in the current events, the dynamic treatment regime. So all those things you can find in my book. So we talk about the semi-parametric foundation first, and then we talk about the classical method, and then we talk about target learning. Okay. So to summary, so today we didn't touch upon the sensitivity analysis, but actually sensitivity analysis to evaluate the impact of assumption because we make assumptions. So we need to make as fewer as possible assumptions. For example, if you are using linear model, you make a linear model assumption, then let's get rid of it. So that's why target learning becomes so important, because we want to get rid of linear model assumptions such that. So Einstein said, make everything as simple as possible, but not simpler. So machine learning is try to make things simpler. So you cannot think, okay, machine learning makes things complicated. No. Machine learning make us to get rid of unnecessary assumptions, like linear assumptions, normality assumptions. We want to use powerful machine learning to get rid of those unnecessary assumptions. And then, like Einstein said, we want to make everything as simple as possible using AI machine learning tools. So I will stop here and thank you for coming today. Let's see if any question. We have 7 minutes left.
Yeah, if any questions that you have, ask Dr. Fang.
So there's a question about I see. Does the attributes in the estimand framework help construct potential outcome? Yes, I think so. It helps us to understand the potential framework, how to use the potential outcome. But in order to construct the potential outcome, we need to rely on our imagination. So we need to have counterfactual thinking. We need to have causal inference thinking. Also, we need to rely on very powerful tool called DAG, direct a cyclic diagram. So DAG will help us to construct potential outcomes. So hope that answer the question.
Hi, Dr.
Okay,
Fang.
another question.
I have a question to. Oh, sorry. Yeah. You can ask someone else question.
I can read the question. Can you provide a quick preview what methods you found most useful for sensitivity analysis? This deserves another webinar because sensitivity analysis is another big elephant. So there are assumptions, 3 assumptions, right? For each assumptions, you need to have one sensitivity analysis method. So we make 3 assumptions here, but actually when you have missing data another assumption, missing random assumptions, you need to have a sensitivity analysis to handle missing data. And now you have in the current events, you have 5 strategies to handle in the current events. Different strategies make different assumptions. So sensitivity analysis, you don't jump to methods. You have to think hard about assumptions. You need to check like here. So what kind of assumptions you make? you make causal assumptions, you make statistical assumptions, you make missing data assumptions, you make indecurable event assumptions. So each assumptions, they are a series of methods you can use. So for example, if you are doing missing element assumptions, so you need to find the methods for missing not element, for example. So if you are doing no unmeasured confounding assumption, you need to find some methods like if there is unmeasured confoundings, what will have happened? So sensitive analysis always goes with assumption. And so basically, so you can, there are another.
Oh, yeah, I'll ask the data, no worries.
Oh, okay. The other just thank you note to me. So thank you for coming, Kai. Oh, there another one. Okay.
Another one.
There are more questions than there. Sorry, so I didn't see that. The other questions said, could you please talk more about how MLDL, machine learning, deep learning are useful in causal inference in last slide? Yeah. So machine learning, deep learning are not making like parametric model assumptions. They are like non-parametric models. For example, boosting and deep learning. So they help us to get rid of linear model assumptions, get rid of logistic model assumptions, get rid of Cox proportional hazard ratio assumptions like that. So machine learning, deep learning help us to make least assumptions. Let's only make 3 assumptions. Let's not make any other linear assumption, normal assumption, parameter. So machine learning and deep learning is very useful nowadays. And then the other question is, how do we do statistical inference when we use machine learning to do causal? Yeah, so 2 methods. Even using machine learning, we are able to develop theoretical formulas. Actually, in my book, we have a method, easy method to develop a asymptotic variance formula. Even you are doing machine learning, doing deep learning, doing random forest, the formula are there. So we can plug in the formula to get the statistical error, standard error. Or you can use Bootstrap. Yeah, Bootstrap is time consuming, but for small sample like phase 2 studies, sample size is small. So we cannot use in the larger sample theory. So you have to use Bootstrap. There's no argument. So if a sample size is small, phase 2 studies, feel free to use Bootstrap. And if the sample size is a little bit large, so you can use in the theoretical formula, which is based on larger sample theory. And the other question is, could you please share slides? I think Deli will share slides to the team. Yeah.
Yes. We are share slide by email.
Betty, so you can ask the question. Only 1 minute left. So sorry to keep you.
Okay, I'll give very quick question to ask. Is that you talk about the 2 methods, one for the propensity score and then the outcome regression. I'm just wondering, does them conflict each other in some scenarios, or they always kind of show you the same?
They are complementary to each other. They are looking at 2 different angles. That's why somehow we can combine them. We can combine them as a double robust method.
Oh, I see. So they always
they are
will
not
show
competing. They are help each other, because that's why we can combine the strengths of both. That's why if you are able to use in a double robust method, I recommend that you use it, because the SAS procedure gives us 2 statements. So you can do the double robust easily, right? Why not? It gives us opportunity to do double robust. Why not take opportunity? Yeah.
I see. Okay, sounds good. Thank you.
Thank you.
Okay.
Thank you all.
Yeah, I think we are exactly on 3 o'clock Central Time. Thank you very much, Dr. Fan, giving this excellent presentation. And I also get educated regarding causal inference. It's very.
You are so nice. Thank you for inviting me.
Sorry, but thank you everybody to attend this webinar. So we'll share the slide and the video. The presentation was recorded, and also when you leave this webinar, we have a survey. If you like to provide some feedback and suggestion to webinar service, please do that. Thank you very much for help. So let's close this today's webinar.
Thank you, and see you.
Thank you. Bye.